Okay, folks, if you don't mind. So I'm going to point out a couple of things to you. I'm hoping that the tutorials, for those of you who had it at 10 or 11 o'clock today, the tutorials kind of supplement and make some continuity with the material that we cover in class. And for those of you who are doing the 4 o'clock tutorial, I hope that you'll also get the same impression. We are going to, as your TAs may or may not have told you, we are going to actually devote an entire session in the tutorial the week of your test to make sure that you're all up to speed. You'll do a practice test in your tutorial to make sure that you're all more or less prepared for the types of questions that we have for you on your midterm. I wanted to go back and revisit a couple of ideas and some concepts, and maybe now in the context of the tutorials, once you've had them, or if you will have them, will make a little bit more sense. Along the way, one of the things that I really want you to get from this concept is that we don't really know a lot about what's happening. We know that the gut actually acts as a barrier, and this will be important, and you should know that it's important. We know that the gut also acts sort of as an immune reservoir, and it's something that actually has immune function associated with it. And this mesenteric lymph node, or the MLN, that I'll go back and revisit with you, is really important. And we know that different antibodies and other things are also produced from the different cells of the gut. So along that line, after that, we don't really understand all of the mechanisms that are involved in things like gut-mediated, microbiome-mediated changes in meta-inflammation. So we have some ideas, we have different models like P75 receptors and knockouts of P75 receptors, things that I'm going to talk to you about today. But the reality is, it's still a very wide open field. The field itself is only a few years old. It's only about 5 years or so, even though we've been influencing our diet for many, many thousands of years. So this lecture, and this week's lecture, actually, because I want you to get ready for the midterm, will be it. That's all that's going to be on the midterm, right up until Friday's lecture, and after that, you should start studying that material to get ready for the midterm. And the following weeks will cover different material related to what I'm going to go through on next Monday's class. We've been influencing our diet in a lot of different ways, and you don't have to know a single thing from this particular slide. But I do want human biology to actually be relevant for you. The types of milk that we drink now are actually all enriched. We don't have pure milk anymore. They're all enriched with a number of different dietary supplements that, in theory, because of research, may improve the function of the nervous system, may improve your ability to increase the strength of your immune system. But the reality is we've been changing all these things without really considering the microbiome. And this is actually the father of our understanding of the gut microbiome. Do any of you in immunology know who this individual is? Any ideas? No? Okay, good, honest answer. So this individual's name is Eli Metchnikoff, and again, don't worry about the names, it's just to give you an idea. He was the first person to think that perhaps by changing your gut microbiome, you could actually change the way your immune system might work, the way in which you were able to process different foods, etc. And he actually was a Nobel Prize laureate, not for the gut microbiome, but he won the Nobel Prize for phagocytosis as a cell biologist. One of his students was actually this individual here, Minoru Shirota, and Minoru Shirota was actually an individual who studied with Eli Metchnikoff, and he came up with this idea that, you know, one thing that we could do is potentially go ahead and seed your gut with new bacteria that is normally not encountered in your gut. And he came up with this drink. Many of you might have had this at some point in time, but again, just to put things in perspective, it's not just here's some bacteria, you're going to eat it and it's going to get to your stomach. There's actually a lot of thought, research, and processes that go along the way to making sure that it gets to the places that it needs to get to. Now, your gut has been colonized from a very, very early time point. And this is a slide I can guarantee you, I've mentioned this to you before, that you should make sure that you know. Now, I went through different aspects of this slide in the last lecture, and we talked about some of the controversies, whether or not the uterine environment in utero while you're in your mother's womb is really sterile, or does it actually contain bacteria? Some evidence, like evidence from the meconium, suggests that maybe it is. We don't see a lot of bacteria in that first poop. We don't see a lot of viral genomes in there either. So whether or not you start off in a sterile environment is still up for debate. However, we know that depending on the type of birth you have, whether it's through C-section or through vaginal delivery, the types of bacteria which you should memorize and which you should know are vastly different. They are not the same depending on your route of birth. Now, I'm going to mention this in passing. One of the things that we know that happens that allows this in the first month or so is that overall we have a decline in what is known as these toll-like receptors, or these TLRs. And these TLRs are going to be something that we come back to several times today. And if you are in immunology or you're taking molecular genetics and biology, you'll know what TLRs are. I'm going to go through this with you in some more detail. The TLR system in early infancy, actually, the responsiveness goes down, which we think allows for different colonization to occur, especially during the first month or so of life. Now, I told you along the way, we have these three dramatic changes. We start off with this population of bacteria, but if you had this population of bacteria now as an adult, over here on the left, you wouldn't be doing very well. And by the time you are 24 months of age, or about 2 to 3 years of age, you now have this stable population of bacteroidetes as well as firmicutes, which you keep as long as you are healthy throughout your adult life. Changing the proportion of these firmicutes or these bacteroidetes will change the way in which your health status changes. And for those of you who have taken the tutorial today, hopefully you have an appreciation of why these things are important, especially with things like C. difficile. But that's sort of the big picture. And again, the one thing that I'm going to point out to you right now is knowing these greenish boxes and the types of things that we start off with is really important. And knowing what we should have, the balance, the proportion between these two dominant phyla is going to be really important throughout your adulthood. And if we change this, then we change your inflammatory status. You might start increasing the production of interleukins or interleukin-1 beta or TNF alpha, and all of these things are important throughout your adulthood. We don't really understand all of that, by the way. We don't understand how, because normally your gut has tons of bacteria living within it. We have trillions of bacteria living through it, throughout your gut. And again, from the last lecture, you know that the place that we have the greatest diversity is actually in the large intestine, in the colon. From the stomach all the way through the small intestine and the colon, we see this gradient, increasing numbers and increasing complexity of these different bacteria all the way through. And so when you are living with this much bacteria in an internal environment, your body has to be able to adapt to this. And we think that some of this adaptation is useful, and some of this adaptation may actually be at the cost of your health. Now, I mentioned to you that this firewall, which represents everything that's on this particular diagram, and I'm going to point out what you need to make sure that you know from this diagram. So I don't want you to be worrying that, oh, you know, is this tiny little detail that wasn't covered on the slide going to be on your test? I don't want you to worry about that. The major points are here on the side, and I want to go through each of these in turn. These commensal bacteria, they're your normal bacteria. They're the ones that you have in normal health. They probably consist of both bacteroidetes as well as firmicutes. And these commensal bacteria that you normally have in health are sitting here in this area, which is known as the lumen, the center region, the opening, if you will, if you cut through the gut and you took a look at it, the donut hole around it. And this is the rest of the surrounding tissue. So if we were to take a look, we know that these epithelial cells will actually produce this mucus barrier. This mucus barrier is going to be more important than you give it credit for. And specialized cells, which we're going to learn in a moment, goblet cells, and I'll introduce this to you now, are going to be important. But in addition to that, other cells here not only produce mucus, but also produce antimicrobial proteins. They basically inhibit the growth or the overgrowth of different bacterial species, etc. However, once in a while, some of these bacteria, for whatever reasons, are able to cross this barrier. They get through the mucus, they get past the antimicrobial proteins, and then these commensal bacteria get inside the tissue itself, inside the mucosal tissue. And once they breach this, there are a number of internal defense mechanisms that make sure that it helps to control this. So wherever possible, we have these different macrophages, these resident macrophages, as well as these dendritic cells. If they happen to encounter any of these, we'll try to destroy these right away or eliminate them as quickly as possible. However, some of those antigens, some of the proteins that are found on the surface of these different bacteria are also going to be captured by these dendritic cells, which will then take them to these mesenteric lymph nodes, these MLNs, these mesenteric lymph nodes, which then help to direct an immune response to the appropriate bacteria that they've taken up. Okay, do you have any questions about this before I go on, or anything that you want to double check? Do I need to know this? Do I not need to know this? Okay, so any of the details I haven't covered so far, you don't need to know things like interleukin 10 being secreted from resident macrophages. We'll get back to those later on. For the time being, I've just pointed out the different cell types and how they bring them into this mesenteric lymph node, or the MLN. Any other questions before I go on? So this is sort of your mucosal firewall protecting you from this bacteria, which is beneficial, but if it's inside your bloodstream, it becomes less beneficial and it becomes a danger. So in addition to that, the presentation of these normal bacterial antigens, these commensal bacterial antigens, by these dendritic cells also help to prime both the cellular responses in immunity, including the regulatory T cells, or the Treg cells, the T helper cells, or the Th17 cells, as well as the antibody-producing B cells. So we have both a cell-specific response and a humoral response. We produce antibodies, and I'd like you to make sure that you know that in response to a bacterial challenge, your body produces IgA-type antibodies from these B cells because they've encountered these particular commensal bacterial antigens before. And part of the reasons why we do this, your gut actually has a lot of immune tissue associated with it. So what are these immune tissues? One of these immune tissues are these mesenteric lymph nodes, or the MLNs. And another one of these is actually known as Peyer's patches. And this is where we have a bigger concentration of these different cell types, the beta cells, etc. So both Peyer's patches as well as these MLNs help to make sure that your gut, the tissue of your gut, actually has an immune function built right into it. Do you have any questions before we go on? Okay, so we don't want this overactivation. We don't want any pathology occurring. And so these are the mechanisms that are at play that allow us to control, to modify, modulate the responses in case any of these normally occurring commensal bacteria happen to cross. And I'm hoping that you pay particular attention to this slide, right? Yes, no? Can you guys actually read any of this stuff? From the back, can you read what's at the very top? No. Yes, this is an Easter egg, and this is one of those things that may or may not be on your slides, but it is on this slide. And I can tell you that there are going to be elements of this slide, this particular slide, that you're going to see on your midterm. Not going to tell you what, but it is there. Yes, it says Easter egg. It says something, something, something. This slide is important, something, something. Yeah, so now you're going to go back over every single slide and go, I wonder if there's something hidden there. You shouldn't do that. Okay, so I also talked to you a little bit about this. So that's the normal bacteria. That's normally what's occurring. We have this balance between these firmicutes and these bacteroidetes, the firmicutes being not so good, the bacteroidetes being pretty good for your overall health. And we know that once in a while, for whatever reasons, in disease cases like Crohn's, or if you're on antibiotic therapy, some of that balance that we have, that we started off with when we were 2 years old, all the way through adulthood, gets out of whack. And when it becomes out of whack, then bad things can happen, because other commensal bacteria, all of you in this room have many different types of bacteria living in your gut. Some of you will have different types of Clostridium or C. difficile that are also growing in your gut. Is that a problem? It is not a problem, because all of the other bacteria normally help to keep it in check because they're nice and healthy. However, if you've suddenly wiped out whole populations of bacteria, either because of a disease state or because you've taken antibiotics, whatever the case is, these Clostridium can start to multiply incredibly quickly. And when they start to multiply really quickly, they will actually induce an associated disease state, which is really, really painful and is really, really bad. It's not easily curable, by the way. This C. difficile releases a toxin which causes the bloating effect in your gut. So it feels like you have like a full intestine all the time. It's not a very pleasant experience. It'll also produce diarrhea, and you're never sure, like, why am I getting all of these different symptoms? We know that if you try to treat this, you try to kill this with other antibiotics. So you have C. difficile, you have much more than you normally should, and now you try to wipe them out in a traditional, conventional way by using vancomycin or other antibiotics, that it doesn't help. It becomes really, really resistant to these forms of antibiotics. And what that means is, if we use the same principle as we've learned before, that oftentimes we have to use a transplant. So we need bacteroidetes. We need normal bacteria to come in and to suppress the overgrowth of the C. difficile. And it turns out that when you do this, instead of using antibiotics, that this method actually turns out to work better. And these other approaches of using active bacteria ingested in the form of probiotics, or giving the supplements that allow your endogenous commensal bacteria to grow in the form of prebiotics has been used. But to date, the best sort of success has actually been an oral fecal transplant. Now, some of you will have heard about C. difficile again earlier today in your tutorials, and some of you will hear about C. difficile again later on in your tutorials at 4 o'clock. This slide is something we didn't take a look at in the last lecture. So does this transplant, this oral fecal transplant, or any kind of transplant of bacteria work for everyone? It turns out that it doesn't. We don't understand all of those processes yet, but we know also that it doesn't we know that there are some factors that likely play a role. So diet certainly plays a role, right? And that's the second part of today's lecture. The type of diet that you have will ensure whether some of these bacteria survive well or not survive well. We also know that there are some genetic roles. We don't understand all of the genetics, that for whatever reasons, that in different offspring, we often find the same bacteria, which means that they're more likely. There's a genetic role that plays some aspect of why we have different types of bacteria growing in different types of individuals. However, we also know that if you took the same offspring, that there's also a unique fingerprint to the different species of bacteria that are growing in them as well. So there might be some uniqueness associated with all of them. Again, the immune system plays a huge role in the gut microbiota. The role of these toll-like receptors that I mentioned very briefly, that in early development, the toll-like receptors very briefly lose their function for about a period of 1 to 2 months. Your immune system allows the bacteria to grow, which it should, and we don't really understand the full sort of implication. What's the What's the back and forth between the bacteria and these toll-like receptors? Yes, early in development, you don't want the immune system to wipe out the gut microbiome, so the toll-like receptor expression and function has to decrease to allow the bacteria to grow. But is there really a balance with the immune system? And again, it's something that we're going to look at very briefly in the next few slides. We know that how fast your gut moves, the rate of peristalsis, and have all of you heard this term peristalsis? The contraction, the rhythmic contraction from the mouth all the way down to the anal canal of the GI tract is known as peristalsis. It's like watching an earthworm move. That sort of motion is very similar to peristalsis. We also know that there are stem cells which exist in the crypts of the um, epithelial lining that allow for new cells to be born. And this also determines whether or not this transplant is going to be successful. We don't understand those roles yet. We just know that the length of this migration is really important. And of course, the different types of mucus. Some of you will have very, very different muck genes that produce different mucin proteins, and you produce very different types of mucus, which allow some bacteria to adhere, some to be repelled, etc. And so we know that even physical factors like mucus play a big role in whether or not your transplant is going to be successful or not. I did this sort of as a joke for all of you, but it turns out that this is going through clinical trials, that those individuals who have not had any success with different types of antibiotics, when everything else fails and you just really feel bad, it becomes debilitating that you can't actually go to work, you can't sit in the classroom, you can't study like you normally would, that if you're looking for options, this oral fecal transplant where you take these freeze-dried fecal samples from a nice healthy individual, and this will become important in today's talk, these human trials are not just for the C. difficile individuals, but we're also looking at it as a form of weight loss. So if your gut microbiome actually plays a role in metabolic syndrome and weight gain, then putting your gut microbiome back into balance, whether it's through yogurt and probiotics or eating someone else's poop, completely logical in terms of our understanding right now of how the gut microbiome works. As I mentioned to you in last week's class and the week before, we don't understand all of the factors that are involved in this. We know that meta-inflammation involves different cytokines. What the source of these cytokines, where they're coming from, what stimulates what cells are not really fully understood by everyone right now, and it's not fully agreed on by everyone right now. But we know that there are certain markers of meta-inflammation that go up, especially if you have either dysbiosis, whether you have metabolic syndrome, or a combination of the two. And these are things like TNF-alpha, the interleukin-1 beta, and that chemokine CC motif ligand 2, or CCL2. We know that your other immune cells, your mast cells, those T cells that I mentioned, the Treg, the T helper cells, resident macrophages, all of these things will also be upregulated when you have dysbiosis, if you have inflammation, as well as having maybe obesity. Again, good bacteria like bifidobacteria has been shown to decrease all of these inflammatory markers, decrease the appearance of these macrophages as well as T cells. And we also know that we can do the same thing in different obese mice models by simply reducing the permeability of the gut wall. So that gut wall, that barrier, that firewall that we talked about, if it turns out to be leaky for whatever reasons to things like lipopolysaccharides, which I'll get back to in a moment, if it's leaky to that, then actually it might be a predisposing factor for the obesity as well as for inflammation, etc. And whether it's this gut dysbiosis that starts that process or it's a consequence, we're not really sure. So before I go on, I'm going to actually take a little bit of a detour. I'm going to go through two slides, which are important slides for you to go back and review for multiple choice. I'll be completely honest with you. I'm not going to be asking you these diagrams for you to come back and draw these diagrams again or label these diagrams. But for multiple choice, I would suggest that these are good slides for you to come back and take a look at. So in today's paper, I'm going to introduce to you the concept of a germ-free mouse. These mice are ones that are bred and born to be used experimentally. They're not these wild mice that you find out in the field. These are found in different labs. We can raise them so that in their gut, there is no bacteria at all, nothing. They are completely germ-free, okay? Not just in the gut, but everywhere else. So is being germ-free good, right? You'd think, well, germ-free, no bacteria. That must be a great thing to have. But physiologically, it turns out that it's not such a great thing to have. So one of the things that we know that happens is that in a germ-free mouse, if we were to take a look at their gut wall, and here it's thrown into these villi, these long finger-like projections are known as villi, and these are found in different parts of the GI tract system, that if we were to take a look, the mucus layer is actually much, much thinner. They don't produce as much mucus. Okay, in these germ-free mice, no bacteria growing in their gut at all. They have no commensal bacteria, unlike these conventionally or normally raised mice that we use in our experiments. And they have all sorts of these different microbiota. They have all sorts of these different commensals, and you can see their mucus lining is actually quite thick as well defined. In addition to that, the mucus itself is different. It's not as adherent. The properties of the mucus make it less likely to be able to act as a physical barrier for any bacteria that they do happen to encounter. In addition, if you take a look, the physical structure of the gut itself is changed in the gut of a germ-free mouse. These villi, 3 of these villi being shown here, 3 villi shown here on the right, these villi are actually thinner. They're not quite as thick. And we also see fewer blood vessels associated with them. They don't have a rich vasculature. They don't get as many nutrients. And all you're doing, the diet is the same, everything else is the same. They just have none of the bacteria that we would normally associate with these mice. The other thing that's a little different about this in this particular slide is that there are more of these stem cells that we find within the crypts of these mice that are conventionally raised. For whatever reasons, we don't see as many stem cells in these germ-free mice. Keep all of this in mind because we're going to talk about germ-free mice in that fiber paper in a few moments. Do you have any questions about this before I go on? Or is there any part of the physiology that I can re-explain to you? Do you have a question? And you have to speak up just a little bit. So these are the villi, and then going down into the epithelial folds are these crypts. So this represents sort of an infolding or an invagination. And those are the crypts. These are where usually you start off having these stem cells. These stem cells kind of migrate and they move upwards. Okay. Any other questions? Yes. Sorry, is the composition of the mucous membrane, is it less adherent? Overall in the germ-free mice, yes. Yep. It becomes less of an efficient barrier. It's less of an efficient barrier to bacteria. So again, we won't worry about all of the details on this slide. The ones that I'm going to point out to you are the most important ones. As I mentioned to you before, your gut actually has immune function associated with it. So we know that you have cells, Treg cells, T cells, T helper cells. We know that B cells are involved. We get these dendritic antigen presenting cells. We have resident macrophages. Your immune system, because it's exposed to so many different bacteria, has to be well developed within the gut. However, we know in germ-free mice that the Peyer's patches as well as the mesenteric lymph nodes are not quite as developed. So you can see here that they're quite small. These little MLNs, the mesenteric lymph nodes, are smaller. They're not quite as robust as we would find in these normal conventional mice on the right. We also know that the epithelial cells themselves that produce these antimicrobial proteins don't produce as much. The antimicrobial proteins themselves are also going down. And we know the beta cells are also, or the B cells are not as responsive. The IgA production by the B cells is also not going to be as high or as robust as we would find here in these conventional mice. So again, smaller Peyer's patches, smaller, less active MLNs, less production of antimicrobial proteins, less production of IgA from the beta cells. Let me just go back to this one slide, and I have to apologize. I had said that I would show you this earlier on. The mucus that we produce is actually produced by a specialized type of cell, which is known as a goblet cell. And this goblet cell is something that I'd like you to make sure that you're familiar with in terms of mucus production. So where does that mucus come from? It's really the goblet cells that produce that mucus. They're scattered throughout the entire villus. They're scattered throughout the entire epithelial layer. So why is this all important? So this is important for a couple of reasons. We have an immune system. That immune system is there for a variety of reasons, probably to keep all of this bacteria in check so that it doesn't become dangerous, because you're in contact with this whole host of bacteria on one side of the lumen, and on the other epithelial side, you don't want those bacteria crossing over. But what happens if they do cross over? And what are sort of the responses that might happen as a result? Well, we know that these different lipopolysaccharides and polysaccharide A helps to enhance the immune system within normal conventional mice. The ones that are normally exposed to bacteria are the ones that have these Treg cells that are already active, ready to go, and ready to produce an immune response. We know that these T helper cells are also ready to go. But we don't really know if this is something that is important in control, or is something that gets changed when different types of bacteria tend to dominate over others. And this is where we're still learning a lot about the immune system and the immune status of mice in this overall context of the GI tract. So we have an immune response. We need that immune response. We don't understand why it seems to be primed and ready to go to deliver these immune responses. But I'm going to take, again, one more segue to hopefully bring some of this into focus for you. There's one hypothesis that suggests that this whole meta-inflammation is thrown off, like our whole metabolic obesity, all the downstream factors that result, getting fatty livers, having larger waists, inappropriate accumulation of fat, all of this might be due to the fact that that gut firewall that we just spent a few minutes going over, if it becomes leaky and if it becomes overly activated, and if some of the different bacterial proteins like lipopolysaccharide or polysaccharide A, or LPS as I've indicated here, if some of that actually goes across the entire membrane and goes across into the bloodstream, then it might actually induce this meta-inflammatory response. Now, I don't need you to memorize all of the details that are on this slide. And for the time being, I'm going to point out a couple of things to you. The one that I want you to spend the most time on is this one here, which is TLR5, which is toll-like receptor, or TOLL dash like receptor, TLR5. We have many members of the family that you see here. If you're paying close attention, and I'm just using this in broad strokes, these are the receptors that are on cells to help identify if there is an infection, whether that infection occurs because it's in contact with a structural element of bacteria, like the flagellin protein in bacteria for TLR5, or if it's in contact with the CPG or methylated DNA for different viruses, etc., or lipopolysaccharides. Whatever it is, this system is designed to recognize that you're being attacked by viruses or through bacteria. In particular, I'm going to pay attention to this TLR5. All of them work in the same way. It doesn't matter if it's TLR1 or TLRX or TLR2. All of them work in the same way. that when the ligand, whatever it's designed to recognize, whether it's the flagellin protein on bacteria or it's a peptidoglycan, whatever it is, when it binds to this toll-like receptor that's found on different cells, it will actually initiate this signaling cascade. It's a sentry system, in other words. And when that happens, one of the things that gets turned on is this MYD88. Here it's abbreviated as MYD88, so I'm just going to call it MYD88. And this MYD88 then accumulates other proteins around it, like the IRAC, TRAP6. And this is all important because one of the things that it does is that it will turn on the transcriptional activator known as NF-kappa B. Now, why is NF-kappa B so important? That cell has just recognized that there's a bacteria floating around me. I've just detected a flagellar protein, which normally I'm not encountering. Now I do. I've set off this chain reaction. And why is that important? Because this NF-kappa B that gets turned on as soon as you detect this flagellar protein then starts to produce those inflammatory cytokines. Now, again, in terms of immunology and in molecular genetics, this is important. This is great stuff. But how is this at all implicated in what we're talking about here in terms of the microbiome and metabolic syndrome and inflammation, etc.? And again, in case you were wondering, I'm not going to cover this over here on the right. This is sort of the overall structure of all of these different proteins. If you want to study it yourself, that's fine, but you're not going to worry about getting tested on this at all. So I'm going to, again, use this in the framework of why did we study TLR5? Yes, you need to know the pathway. Yes, you should understand how MYD88 gets turned on and how turning MYD88 might induce an inflammatory cytokine response. A number of years ago, some researchers decided that they wanted to look at the role of these toll-like receptors in immunity in a variety of different tissues. And what they started to do was to knock out different toll-like receptors. They knocked out toll-like receptor 1, 2, 3, all the way through. And one of the things that they found was that they wanted to examine the role of toll-like receptor 5. And toll-like receptor 5, or TLR5, normally interacts with the bacterial flagellar protein. So these bacteria don't just sort of go with the flow. They actually move around. They have these little motor proteins, the flagella, which act like sort of a whip-like fashion that allow them to move from one end to the other. And they're found on bacteria. So this flagellar protein or flagellum protein, which is called flagellin, which allows it to kind of whip back and forth, this is normally detected by that toll-like receptor 5. So in other words, if you have a bacteria that uses flagella to move around, the toll-like receptor 5 is going to be the one that detects it. So they knocked this thing out. They wanted to see what is the end result of knocking out toll-like receptor 5. The surprising thing for them was that these toll-like receptor 5 knockout animals, where they assumed that they're only going to affect the immune system, potentially development, started to have metabolic problems. And these metabolic problems are that they weighed a lot more than mice that did not have toll-like receptor 5 knocked out. They also started to not be able to control their levels of lipids in their blood. They had unregulated blood lipid levels. So they started to have increased or elevated blood pressure simply by knocking out a protein that is used to detect the presence of bacteria from all the cells within the body. They also started to develop hyperinsulinemia. They had elevated insulin levels even after a blood glucose challenge. And they also started to develop what looked like diabetes because they started to have slightly elevated levels of blood glucose. These results were exactly not what these scientists wanted. They wanted to know, you knock out this bacterial detector, your immune system is probably going to be messed up. But these mice actually developed metabolic syndrome. All the things that I've just described to you are metabolic syndrome. You weigh more, you have unregulated blood glucose, you have hyperinsulinemia. All of these things are related to metabolic syndrome. These mice also started to eat more food as well. They also started to exhibit insulin resistance, so their insulin levels stayed high even after a glucose challenge. Nothing was affected. And if they started to give these mice that ate more anyways, if they started to change their diet and gave them high-fat diets, they developed a fatty liver. And we talked about the diagnostic criteria of a fatty liver when we talked about the P75 knockout animals a little bit earlier on. So in other words, they had metabolic syndrome by knocking out in all of the tissues and all of the cells this toll-like receptor 5. So why did this occur? So they had no idea. And they started to go back and do a number of things that were not related to immunity. So when they went in and they used high doses of antibiotics to kill off all the bacteria, these are not germ-free, these are conventional mice, they have bacteria growing inside of them. When they use high doses of antibiotics to kill off the intestinal bacteria, they actually found the metabolic syndrome disappeared. So it suggested that toll-like receptors somehow, which they weren't sure of, that somehow were causing changes in the gut bacteria that led to these metabolic consequences that led to metabolic syndrome. So when they started to go back and look, what bacteria did we actually kill off in these toll-like receptor 5 knockout animals? When they started to go back and examine the different populations of bacteria in their gut, what they found was that knocking out toll-like receptor 5 that led to metabolic syndrome and all of the sequelae of things like fatty liver, diabetes, etc., that they saw an overabundance of those firmicutes that we talked about earlier. So the balance between the bacteroidetes and the firmicutes was thrown off balance. And they developed metabolic syndrome by increasing the level of firmicutes, decreasing the level of the bacteroidetes, and if you wiped out everything, the metabolic consequences disappeared. Again, they're not really sure of how all of this is related. They just know that somehow this signaling mechanism is important. So I left you with this, the fact that you are changing in your diet, and coming back full circle, you are changing in your diet. Generationally, you are changing your bacteria. And from generation to generation, at some point, it becomes too late if you do not have a high fiber diet. And I haven't told you what a high fiber diet constitutes. You are changing your gut microbiome, and you are changing it at the level where there is less and less complexity. And again, if you went to tutorial today, one of the things that you learned in your tutorial was that having lots and lots of bacterial diversity is actually good for you. It's like having lots of genetic diversity. You're able to handle things in a much better way than if you have a very small pool. And so I'm going to go through some of this now, and I'll finish on Friday, and that will be the end of the block that you need to study for your midterm, because I want you to start organizing your notes, coming in to see me if you don't understand something, talking to your TAs if you don't understand something. And again, I want you to get the most out of this course that you possibly can. One of the reasons why this paper was important is that our diets, as I mentioned, have changed. Sometimes we're doing it in a good way, sometimes we're doing it in a not so good way. We all know that our diets can be changed through supplements. Having more vitamin D in milk is probably a good thing. Having DHA in our milk is probably a good thing. We don't know how it affects the gut microbiome, but we know that originally the gut microbiota, all of the different bacterial species living in those Paleolithic cultures, the ones that you were reading about and the ones that you will read about, of the hunters and gatherers, before we became an agriculture-based society, we know that their diet was very different than ours. We also know that they probably had a greater diversity of bacteria than we do now. And our Western diets probably have a lot to do with the lack of diversity that we have now in our gut microbiome compared to the early hunter-gatherers or a rural agrarian, or the early farmers, in other words. So how does our diet actually change our gut microbiome? So one of these concepts is that the microbiome, the gut bacteria, has in it or uses accessible carbohydrates. So you eat food, you eat fiber, your body doesn't really process fiber, but the gut bacteria like it. And those MACs, these microbiota accessible carbohydrates, the fibers that are in our diet, are actually used by our gut microbiome. And these are found within certain diets, but they're not found so much in our diets. We have really high carbohydrates as well as really high fats in our diets, and overall we have very, very low fiber levels compared to those early hunters and gatherers. We can still compare our diets, our gut microbiomes, with individuals who are living in rural agriculture-based societies, as well as hunter-gatherers that still exist. And we know that even now, this is still true. It's not historical, this is still going on now. So the object or the hypothesis was, could a change in diet where it's low in fiber, and in other words, it's high in fat, high in carbohydrates, can that actually change the gut microbiota within the gut? So a couple of things to mention to you. So all of these different mice, if you don't know what a humanized mouse is, the way that we do this in terms of our studies here is that you take these mice, and in this case it happens to be a particular type of mouse. It's called a Swiss Webster mouse, and what you do is you give it an oral gavage of human fecal samples, right? Remember those oral human fecal freeze-dried pills? Instead of actually using freeze-dried pills, you put it into liquid form, and then you put it down the throat of the mice. And I'm sure the mice don't really like it, but they don't really have a choice. And so this oral gavage of human fecal samples that were obtained from, and again, I'm using their own words, a healthy anonymous donor, an American male with an omnivorous diet, these are the humanized mice. These are not bacteria that normally grow because of the mice bacteria of the different diet that they have, but these are the human microbiota that came from a human donor. And all of these mice have it, they take it up, and they now have the same microbiomes that you have, and this is why we call them the humanized mice. And they all look like the same phylogenetic makeup. In other words, they have the same bacteroidetes, firmicutes that the donor has. So I haven't mentioned to you what is a high MAC or microbiota accessible diet. So this is where it's rich in plant fibers, rich in plant polysaccharide fibers that the different gut bacteria can use. And it comes from a variety of different plant sources. So things like corn were used, soybean, wheat, oats, alfalfa, as well as beets. All of these were constituted a high MAC diet or a high fiber diet. And a low MAC diet is one where we don't use our carbohydrate sources from these different types of plants, where it's from sucrose, the same sorts of things when you order that double, double, triple, triple, quadruple, quadruple from Tim Hortons. That's that same sucrose that you have being put in there. And cornstarch, again, that sort of high fructose corn syrup that is so prevalent in our diet. Now that cornstarch was also used as well, and a very, very small amount of cellulose, which normally doesn't break down, but is used as a control for fiber. So cellulose isn't something that we can digest well, and even some of the bacteria don't digest it very well. So again, I'm not going to go through all of the details right now, and I'll go through this in a little bit more detail. So these humanized mice that have the same bacteria that we have are given either a high MAC diet with good fiber sources or low MAC diets with low fiber sources. And then they wanted to collect the fecal samples. And you can imagine how great a project that would be. Sit around all day, wait for your mouse to poop, collect the poop, weigh the poop, and then analyze the poop. Great project for an undergrad, right? You'd be dying to do that project. So they do all of these different studies, and what this shows is the diversity, the overall diversity of the gut microbiome. So higher up on this y-axis, which is known just as a principal coordinate, the more diversity you have, the higher your principal coordinate number. If you're on a high MAC diet, you have lots and lots of diversity. You stay on a high MAC diet in green, you get lots and lots of diversity all the way through. You go into a diet switch where you start off all the same, and then you switch them down after a few weeks, you lose a lot of that diversity. You then follow them. When you then put them back into a high MAC diet, as you see over here, do they come back? No, they don't. They stay low. The diversity never comes back after that generation. I haven't shown you another generation or their offspring, but the diversity never comes back. And it's something that was surprising. We'll go through some of the details in the next lecture, but I wanted to just share what a humanized mouse was. You should know what that means. You should also know what this concept of a high fiber diet, what that actually represents, and how many of you would actually like to partake in a high fiber diet? Anyone want some high fiber? So what do you think is high fiber, by the way? Just out of curiosity. What is high fiber? So again, we're rapidly running out of the gut microbiome. So these sorry, and I still like PC products. I'm not part of the Weston family, by the way, in case you were wondering. Is that a really good fiber diet? This says Nutri-Grain. Is this a good high fiber diet? It actually says you get whole grains are in this. Would you rather have that? And let me give you one last one. This is actually Greek yogurt with 2 grams of fiber. Is this the best fiber that you get? Yes? No? Not sure? I'm going to tell you that all of that stuff is completely superfluous. This is actually your best source of fiber, right? Which is actually fruit. Per weight, there is more fiber in this fruit than in any of those. And actually, it turns out that this fruit is more expensive than any of those. So keep that in mind as well. And please help yourselves. I'll see you all on Friday.